Gamma Gummy. Corporate sponsorship provided by Chipotle Mexican Grill. Many of us remember when a few months ago Chipotle was responsible for an E. coli outbreak, but E. coli itself isn't necessarily bad. In fact, E. coli exists naturally in the human intestine. So what made this strain of E. coli so dangerous?
E. coli outbreaks spreading across the country.
Grocery stores have been clearing their shelves of bagged fresh spinach.
The outbreak has spread to at least 19 states, is being blamed for one death and has sickened at least 109 people.
Some strains of E. coli are believed to initiate and sustain infections because of its ability to produce alpha-hemolysin. Natural E. coli occurring in the human intestine do not have this capability. So what is this alpha-hemolysin? Scientists studying the toxic activity of Staphylococcus, a bacterium in the early 1800s, noted the ability of some strains to produce supernatants capable of hemolytic activity or the destruction of cells. Most of these early studies were found lethal in just a few minutes in guinea pigs and rabbits, but were not thought to be lethal in humans. But the protein's tragic rise to fame came in 1928 when 20 children in Bundaberg, Australia were immunized with a contaminated antitoxin preparation. Hours later, 16 of the children experienced vomiting, high fever, unconsciousness, and convulsion. Within 2 days, 12 of the children had died. In the follow-up investigation, Frank McFarlane Burnett from the Royal Commission of the Commonwealth of Australia noted massive production of toxic substances must have taken place as Staphylococci were the responsible agents. Burnett discovered that culture supernatants from the vaccine-contaminated Staphylococci strain caused hemolysis and lethal injury upon injection into rabbits. When Burnett and his team neutralized the supernatant with formalin or derived serum from immune rabbits, they prevented hemolytic activity. Frank McFarlane Burnett would later go on to predict acquired immune tolerance, earning him the Nobel Prize, and developed the theory of clonal selection. In the 1960s, scientists were able to isolate purified alpha-hemolysin from culture supernatants and perform experiments that offered insight into how this toxin achieved its remarkable lysis of red blood cells. A series of experiments sought to measure damage caused by the toxin and found that markers would leak out of cells treated with it. This was consistent with the hypothesis that alpha-hemolysin somehow attacked and damaged the cell membrane. In the 1980s, scientists got their first glimpse of alpha-hemolysin structure under electron microscopy when they saw ring-like structures 10 nanometers in diameter with 6 to 7 subunits and a central pore approximately 2 to 3 nanometers across. The structure was fully determined in 1996 when Song and colleagues solved the crystal structure of the protein. Alpha-hemolysin has a mushroom-like shape and measures up to 100 angstroms in diameter and 100 angstroms in height. A membrane-spanning, solvent-accessible channel runs along the sevenfold axis and ranges from 14 angstroms to 46 angstroms in diameter. On the exterior of the 14-strand anti-parallel beta barrel, there is a hydrophobic belt approximately 30 angstroms in width that provides a surface complementary to the nonpolar portion of the lipid bilayer. The interfaces are composed of both salt links and hydrogen bonds, as well as hydrophobic interactions. 3 domains of alpha-hemolysin have been identified. First, a cap on the extracellular face of the protein is exposed to the aqueous environment. Second, the rim, which rests on the host plasma membrane. And third, the stem domain that forms the membrane-perforating beta barrel pore. Here, alpha-hemolysin is simulated in the lipid bilayer. To summarize, alpha-hemolysin in the bloodstream forms a homoheptameric beta barrel in biological membranes, permitting rapid release of ATP and potassium ions while restricting movement of macromolecules across the cell membrane. An influx of extracellular calcium flows into the cell, stimulating hydrolysis of membrane phospholipids and toxins, eventually leading to cell and tissue death. Robert Tox, from the Centers for Disease Control, has this to say about alpha-hemolysin.
First thing that it does is it starts killing the intestinal cells, and then it gets into the bloodstream. It's like running the blood cells through a Cuisinart or something. They get sliced into little bits, and in a few people it targets the brain.
So how might we treat infection with alpha-hemolysin producing bacteria? Well, alpha-hemolysin membrane penetration is only possible because of ADAM10 or ADAM10, a zinc-dependent metalloprotease expressed as a type 1 transmembrane cellular receptor on a wide variety of host cells. Normally, it is responsible for cleavage of ectodomains of a large number of host proteins, and membrane binding of alpha-hemolysin increases this activity. Recent studies investigating ADAM10 inhibition as a strategy for improving outcomes in alpha-hemolysin-inflicted pneumonia and other illness have shown promise in animal trials. Alpha-hemolysin may be utilized in future biotechnology applications where DNA polymers can be electrophoretically driven through the nanopore of an alpha-hemolysin channel and a lipid bilayer. Recording translocation duration and current flow for traversal of individual nucleotides has been shown as a potential low-cost strategy for analyzing DNA polynucleotides. Anyhow, food for thought when you eat your next Chipotle burrito.